obviously, right, for these,
Priorities.
the bacteria is clearly very resistant to that antibiotic. It's growing on top of it. Come on. It doesn't care. So zones of inhibition, remember that we had to measure. And the reason why we had to measure was because of molecular weight. So think of it as the bigger you are, the bulkier you are, you're not getting very far, right? The smaller that you are, the tinier you are, you diffuse further into the agar. So lower molecular weights have bigger kind of diffusion rates than something that's bigger and bulkier. So that's why we had to measure that diameter to kind of compare to that table to make sure whether it's resistant or sensitive to it. Okay. Okay, beta, okay. Terms, I guess, disinfection, antiseptics. So disinfectant surfaces, antiseptics on tissues, degerming, scrubbing. Okay. Dang, this is long. I totally stopped way earlier last class. I forgot there was like 5 pages left. Okay, so this is on your lab folder. It's called supplemental info. There are a couple of slides that we did, or plates that we didn't do that are going to still be on the test. So MSA we've done, right? Selective is going to be the salt that selects. Differential based on their ability to eat the sugar, which is mannitol. This is a plate from a couple semesters back on another campus. So 1, 2, 3, and 4, you see that only 1 and 2 grew. 3 and 4, they were added to the plate, but they're not halophiles, so they didn't grow, right? So it doesn't matter whether it's gram-negative or gram-positive, it just didn't grow because it's too salty for it. So only 1 and 3 were able to grow. Technically, only 1 should have fermented mannitol, 2 shouldn't have. But this is what these results look like. Both of these are halophiles or osmotolerants. You see growth on both plates, but only 1 is capable of eating the sugar, right, which is S. aureus. So this one's still a halophile or osmotolerant. It just can't ferment mannitol. This was a halophile that can. Okay, McConkey and EMB, they're the same exact plate. They both select for gram-negative. You cannot grow a gram-positive on these plates. They both differentiate based on lactose eating or fermentation. So, same thing, 1, 2, 3, and 4. 1 is our gram-positive. Gram-positive doesn't grow. It's added on there. You might be able to see kind of where it's streaked on the plate, but it didn't grow. 2, 3, and 4 are gram-negative bacteria, but of these, only 3 and 4 are capable of fermenting lactose. So you see a different color. This one, gram-negative, just can't eat lactose. It grows on the plate. It just can't eat the sugar. Same thing for EMB, right? This is contaminated this is gram-positive, it shouldn't have grown. But we've got 2, 3, and 4, right? They're all gram-negative. They all grew on the plate, but 3 and 4 are the only ones that are capable of eating lactose. So they changed it to a black color. In this case, eating lactose changed it to a pink color. The other one is, so we've talked about blood agar. It's enriched and differential. So enriched with blood, differential based on hemolytic ability. Clear, completely lysing it is going to be beta, alpha is partial. So it's got a green tint, and then gamma just grows. All right, gelatinates we didn't do, but this is on the test. So this is an enzyme that digests gelatin, which is a protein that's related to collagen. This is why we don't use gelatin for our plates or our slants, because a lot of bacteria, especially some that can infect us, produce this enzyme. So if we were to plate this bacteria and come back the next class, you'd have a little pool of liquid in the test tube or on the plate. So this is a virulence factor because gelatin is related to collagen, so it can eat gelatin, it can eat collagen. This allows for this guy to not
Sure, please.
Okay, so MRVP, okay, so let's say this is the VP test, which would be a positive result?
The one on the right.
Yeah, red. Let's say this is the MR test, which one would be a positive result?
The pink one. Right? No.
Yeah, red. It's red for both of them, right? Mixed acid, just know the purpose. Mixed acid, don't care if the pH is low. VP, they do care that the pH is dropping, they try to neutralize. Okay, this table, so I guess we're trying to group the organisms based on what they are. Obviously, all negative would be our red test tube. So A would be number 2. Which one do we think E is? 1, 2, 3, or 5? Or 4? Or 5? 3. Why?
Because it produced sulfur.
Yep, exactly. So E is 3. What would we call D?
5.
5, yeah. Because the hint is gas. I don't know why it's got dextrose instead of glucose. I don't know. This is weird, but this would be D. B would also be our negative because it's orangish, reddish. And then obviously the one that only ate the sugar on the bottom is going to be number 1. Didn't eat the sucrose or lactose in this one. Alright, so remembering that molecular weight influences how far it diffuses. The higher the molecular weight, the lower the diffusion, I guess, across the agar. But either way, this one's clearly resistant, right? Alright, this is what that EMB plate looks like. So what would we call this, guys? Is it gram positive or negative? It's growing on it. Negative, gram negative. So only gram negatives will grow. It inhibits gram positives. The color is black, so you can see it's kind of leaching into the agar, so it's changing the color. So is it capable of fermenting the sugar?
Mm.
Yeah, it ate the sugar. Otherwise, it would just grow on the plate, right? What test is this?
Urease.
Urease. What's a positive one? The bright pink, right? So, and the enzyme does what? What does it produce?
Ammonia.
Ammonia. Alright, what type of hemolytic activity is A? Beta, yeah, beta. So remember, complete lysine will turn that blood clear. You can see right through it. B, so that it was a trick question, because you see the A and the B. But B is what?
Alpha.
Alpha, so it's partial, so you get a greenish tinge, and then C is what?
Gamma.
Gamma, just gross. Alright, so this is an MSA plate. What organisms are halophiles or osmotolerant? Yeah, definitely not E. coli. So you see where it's streaked on there, but he didn't grow. So only the 2 top ones have colonies, and the other one that we can't see its name. So the top colonies, right? E. coli, it was too salty for him to grow. Which one is capable of fermenting mannitol? Yeah, Asorius. The other one's still a halophile, but it just didn't eat the sugar. Alright, which one is positive for amylase?
B.
A or B?
B.
B, right? Amylase ate the starch. There's no more starch for iodine to bind to, so you got a clear area around the colony where the bacteria ate the starch. What enzyme are we testing for here? Huh? I'm hearing, I can't hear. I think this fan is going. What enzyme? Starts with a C. Catalase. So catalase, we poured hydrogen peroxide. What is the bubbles that we're seeing? What does catalase turn hydrogen peroxide into? Water. I heard water. And these are the phenol red broths. What's the little tube that's inverted called?
Durham.
Durham tube, and it traps CO2 gas. So which one's positive for CO2 production?
The yellow one.
Yeah, the yellow one, right? So maybe these two are positive for sugar fermentation, these clearly are not. When it gets hot pinkish, it might be even protein catabolism, right? It wants to keep living, it starts breaking down proteins for energy. Alright, so this is our sim. Which one is positive for hydrogen sulfide?
The last one?
Yeah,
I mean,
with the black, right? Which one is positive for indole or the enzyme tryptophanase? The cherry red rings, right? So 3 and 4. We can't tell motility, but let me see. I guess I can draw. Alright, so this one stayed just like this. This one, we have our little line, but it's kind of like this. And then this one is just all over it. Alright, so which ones are modal?
Second
2
and
and
third.
3, yeah. 2 and 3. So even if it's just, it moved a little, the fact that it moved, right? Alright, so which one is positive for citrate permease or citrase?
B.
B, right? It turns a blue color. Oh. Alright, which one's positive for gelatinase?